To explain the resting potential of the cell membrane, it is better to first deal with a simple example of how charged particles move through a semi-permeable membrane. In this example, an environment is considered that is divided into two parts by a semi-permeable membrane. This membrane is only permeable to cations, while anions cannot pass through this membrane. Cations diffuse from part A to part B in the direction of their concentration gradient. The remaining negative charges on side A and leaving of positive charges from this side cause the membrane to be negatively charged on side A and positively charged on side B. At this time, the concentration gradient causes the cations to be attracted to side B, while the electrical gradient causes the cations to be attracted to side A. Finally, the combination of both chemical and electrical forces equilibrates the membrane electrically. Here, the net diffusion of cations becomes equal to 0. Now, these ions have a potential that is called the equilibrium potential. The equilibrium potential for each ion can be measured by the Nernst equation and is usually expressed in millivolts. The concentrations of different ions inside the cell are different from those outside the cell. In general, most neurons have higher sodium and chloride ions levels outside the cell than inside the cell, while the concentrations of potassium ions and negatively charged proteins in the cell cytoplasm are higher than outside the cell. Ions are charged particles, so they cannot cross the cell membrane's lipid bilayer. The passage of ions is done by transmembrane channel proteins and transporter proteins. The net movement of ions can make the cell membrane electrically charged. The resting membrane potential is the difference in electrical potential between the two sides of a cell membrane in a resting state in all living cells of the body. The resting membrane potential of neurons in physiological conditions is about -70 millivolts. Several ions are involved in creating the resting potential on the cell membrane. The resting membrane potential can be calculated by using the Goldman Hodgkin Katz equation. The resting potential of the neuronal cell membrane is very close to the equilibrium potential of the potassium ions. Because the cell membrane is more permeable to potassium ions in the resting state, and these ions can diffuse from inside the cell to outside through the leak channels. But this membrane has a little permeability to other ions. For example, very small amounts of sodium ions can diffuse from outside the cell to inside. However, the operation of the sodium-potassium pump prevents excessive accumulation of the potassium ions outside the cell as well as the sodium ions inside the cell. Therefore, the differences in ions concentrations between intracellular and extracellular space and the high or low permeability of the cell membrane to different ions are two important factors in determining the resting membrane potential.